get started then. Thanks for coming to Robbie's finishing talk. It's a great pleasure to introduce him for near the end of his time as a PhD student in our department. It's been great to have him in the lab. Robbie's a native of Indiana, and it's great to see his parents here as well to come and find out what he's been up to for the last few years in my lab. And what he's been doing is studying an amazing light-driven enzyme, photosystem II, and trying to figure out how that enzyme gets made in Chlamydomonas in particular as a model organism. So he started with a collection of mutants that we have that affect photosynthesis, found this mutant that couldn't make any photosystem II, and then figured out what was wrong with it, and it turned out to be kind of a surprising gene because, as I'm sure Robbie will explain, we thought it was doing something else. And he was very thorough about this. He studied the function of this gene not only in Chlamydomonas but also in cyanobacteria and plants, so he became an expert in the molecular genetics of several photosynthetic model organisms. And then he'll tell you about his clever approach to try and figure out what this thing is actually doing when you have a mutant that doesn't make any photosystem II. It's kind of hard to figure out what's wrong with that photosystem II, so he's got a really clever approach for addressing that. Beyond the excellent science that Robbie has done, he's been a really valuable member of the lab. He has a great sense of humor. And I think everyone knows, what a great member of the department he's been. He's done a lot of service for the department. He's a TA for my class. He's just a really valuable member of the lab who organizes our small group meetings and organizes our NCAA tournament pool every year, which is probably the most important thing that he does. So Robbie, congratulations on reaching this milestone.
Thanks again for showing up. I appreciate it. It's a Friday afternoon. A lot of you guys probably are ready to leave. But like Chris said, yeah, I study photosystem II assembly, and that's sort of why the title is having to do with photosystem assembly. I don't think I need to say a whole lot more about that, but yeah. So let's get into this, I guess. So I wanted to start with something that many of you probably aren't sort of aware of. But photosynthesis, as it turns out, is pretty important. And I've shown here, that's true. So these are photosynthetic organisms you can find all over the world, different parts of the world. A lot of these are from California, but you can tell there's an amazing diversity of photosynthetic organisms on the planet. And these guys all have one thing in common, which is that they use photosynthesis as their sort of predominant form of life, or predominant form of metabolism. So all of these guys are taking light energy and turning it into chemical energy and using that chemical energy to fix carbon dioxide via the Calvin-Benson cycle. So if you take home one thing from this talk, that would be a nice thing to take home if you don't already know it. So when we talk about photosynthesis, which we do a lot in Chris's lab, there's a couple main flavors of photosynthesis that we like to talk about. But the main one is oxygenic photosynthesis. And this is the one that probably most of you are familiar with. And that's going to be the one that I talk about for the rest of this talk. But I just want to give a brief introduction to this, because again, not everybody sort of stares at these things all day. But basically, when we talk about photosynthesis, it's happening inside of a whole bunch of different organisms, from plants to algae to cyanobacteria. And what I've shown here, I think is Chlamydomonas, probably. It's this unicellular green alga that I'll talk about later. I should probably know this. And you can see here this sort of green, gooey blob. And if we are to cartoonify this green blob, you'll see that it's a chloroplast. This is an organelle. It's derived from an endosymbiotic cyanobacterium that was swallowed by this eukaryotic cell a long time ago. And you can see here these green pancakes, just the scientific term for them, I believe. They are membrane stacks that actually contain all of the protein complexes required for photosynthesis. And these guys, if we again zoom in even further, are listed here. These are the major players of photosynthesis. And again, this is way oversimplified. They're not just blobs, obviously, but what we have is photosystem II, which I'll talk about later. This cytochrome B6F complex, which is really important as well. They're all pretty important. Photosystem I, which is another photosystem. And then this big ATP synthase that actually uses a pH gradient to drive the formation of ATP, or chemical energy in the cell. So let's zoom in a little further. This is about as much as I'll zoom in, don't worry. To photosystem II. So this is my sort of cartoon representation of photosystem II. It's the best of the two photosystems by far. And it's really special because it actually uses light energy. So it has chlorophyll, which is this green pigment that you guys all see every day and probably maybe don't appreciate so much like we do in Chris's lab, but it harvests light energy and uses that to split water into oxygen. And this is an incredibly difficult chemical process. It's very difficult energetically. So photosystem II is particularly incredible because it's able to use light energy to do this. And it's this renewable source of energy, water, and it's releasing oxygen, which all of us breathe every day. It's composed of between 20 and 30 protein subunits, so it's a pretty complex enzyme. And it has tons of different cofactors, some of which I've shown here in sort of cartoon form. But these include chlorophyll, like I mentioned, heme, which a lot of you are probably familiar with, as well as a lot of metals like manganese, calcium, iron, all of that fun stuff. And one of the sort of outstanding questions in our field is how this complex gets put together, right? I mean, you can see here, in cartoon form, it even looks pretty complex, and in like a molecular or atomic structure, it's also extremely complex. So the question that I had sort of when I joined Chris's lab was, how is this complex machine built? And we can get some sort of clues from that just by looking at how humans build complex machines. So we know that a car is an important machine, and it comes from all of these sort of component parts, right? You have your frame, you have your door, your window, your engine, all your tires, all of that stuff. And I don't think any of you in this audience think that those just enter a factory somewhere and then a car is spit out, right? That's ridiculous. So what you have to build a car are assembly workers. So these guys right here, this guy with the yellow hat, maybe he puts the engine in the car. This guy with the red hat, maybe his job is to put the tires on the car. And this theme of assembly factors or assembly workers is conserved in Photosystem 2 assembly too, not surprisingly. So you don't just throw in all of these things that make up a photosystem into a cell, wave your hand or wait, and have them all come out as a nice photosystem that's oxidizing water and making oxygen. You need assembly workers. So these guys are the ones that I've been studying for the duration of my PhD. So again, if you could summarize my PhD in a slide, which please don't do, that makes me feel really sad about all the stuff I've done. And basically, who the hell are you guys? What are you doing around here? And the one that I'm going to focus on is Red Hat over here, or green shirt, Red Hat, I guess to be more specific. And his name is RBD1. His order, I don't want to be sexist. It's the important enzyme or protein that I've been studying throughout my PhD. And so if I were to split this talk into two parts, which Chris has nicely already done for me, it's the isolation and characterization of this mutant, which lacks RBD1, and then this, clever was his word, not mine, approach to sort of study the function of this protein in vivo. So let's dig right in here into this TUPAC mutant. So Chlamydomonas is the main model organism we use. I don't want to offend all the other people in our lab who study different organisms, but it's the bread and butter of Chris's lab. It's a unicellular green alga. I might call it clammy later in the talks. Don't be shocked or surprised by that, I guess. It's haploid, so it predominantly lives as a haploid organism, which is really useful for doing genetics. It also has a sexual life cycle, which is an extremely valuable tool, as some of us in the lab are finding out with sort of non-sexually reproducing algae, because you can take two mutants and you can mate them together and get a double mutant out, which really helps you do a lot of things. But the most important or the most exciting part of this little green blob over here, for me at least, is that you can grow it in the dark with a carbon source in the medium. So you can knock out photosystem genes, photosynthetic genes, and it's still able to grow because you're basically feeding it sugar, which is a super useful tool for studying photosynthetic mutants. You know, in a Arabidopsis or a plant, for example, if you were to make a mutant that doesn't have photosystem 2, it would probably die very, very quickly, and in fact it does. So this ability to use non-photosynthetic mutants is extremely important. And as Chris mentioned, when I joined the lab, there was this enormous collection of Chlamydomonas mutants that were absolutely requiring this carbon source in the medium. And so I screened through them to try to find these sort of photosystem 2 assembly component mutants. So when I searched for second photosystem assembly component mutants, which I named TUPAC, which apparently is a rapper also, I found one in particular that I was interested in. And you can see here, so this acetate is the carbon source we use. It's the thing that sort of allows this guy to grow non-photosynthetically. And so when we spot all these little algal cells onto a plate with acetate, you can see our wild type grows totally fine. And our TUPAC mutant doesn't seem to grow as well, but it still survives. And these are grown in light, so keep that in mind. But if you actually remove the carbon source in the medium and you try to grow them, you can see that TUPAC dies. And the wild type grows totally fine. So when I screen for these mutants, we use this parameter called variable fluorescence, or FV/FM. I'm not going to get into the details of it. If you want to ask later, feel free. But it's sort of a proxy for photosystem 2 activity. We use it to sort of know how much photosystem 2 is active in the cell. So when we look at this TUPAC mutant, we can see it has no photosystem 2 activity. There's no FV/FM, no variable fluorescence, but our wild type, again, is totally fine. So this sort of pointed to the fact that TUPAC might have some sort of specific PS2 deficiency. And if we go ahead and assay at the protein level what's going on inside the cell, we can see that indeed photosystem 2 protein subunits are specifically decreased. So these guys right here, D1, D2, and CP43 are all parts of photosystem 2. They're all things that make up the complex. And you can see the TUPAC mutant here has very, very little amounts, or low amounts of these protein subunits relative to our wild type. Whereas photosystem 1, that's what these two are, cytochrome B6F and the ATP synthase are all relatively unchanged. So the surprising thing here was that this 2POC mutant had a photosystem 2 specific defect with all other protein complexes more or less unaffected. And we have some other biophysical measurements in vivo to back that up. I don't want to bore you with the details here, but you can hopefully trust me on that. And you may have also already noticed, or paid attention earlier, that this RBD1 protein is missing from 2POC. So we did some work genetically to figure out what was going on. It turns out RBD1 is absent. So if you clone the gene, or I guess when I clone the gene for RBD1 and I put it back into 2POC, we restore photoautotrophic growth, so these guys can do photosynthesis again. They grow without acetate. And all of these photosystem 2 protein subunits levels are restored. So you can see here very clearly at least D2, D1, CP43 are back up to wild type levels. So that told us that this RBD1 gene is apparently required for photosystem 2 activity. Okay, so you might be asking, what is RBD1? It's a fair question. RBD1 is a 16 kilodalton thylakoid membrane associated rubidoxin, which is a mouthful and basically a complicated way of saying it's a small protein that's in the chloroplast and it binds iron. And you can see here that this rubidoxin domain, which is in red, labeled rubidoxin, has these 4 conserved cysteine residues that actually form this nice little conformation that binds one single iron atom. It has this blue domain, membrane anchor, that allows it to associate with these little green pancakes that I showed earlier. And it has this transit peptide that sort of directs the protein into the chloroplast where it can function. And rubidoxins have been known for a while, as Bob knows as well. They were discovered here at Berkeley, and they're known to participate in sort of a wide variety of processes in bacteria and archaea, including some of the things that I've listed here. But as for what they're doing in Chlamydomonas, we aren't really sure. We think it's obviously photosystem 2 assembly, but that still remains. Specifically, what it's doing is sort of up in the air. But the interesting thing, or I guess a lot of things are interesting, but this is interesting to me, is that there's this membrane anchor right over here. And if you look into the literature, or if you look into the databases, almost all rubidoxins that exist don't have a transmembrane helix. They exist only as this sort of soluble rubidoxin domain. So this one in photosynthetic organisms seems to be unique. And it turns out that every single thing that does oxygenic photosynthesis has this rubidoxin in it. So it's been conserved over billions of years of evolution. It's extremely important, apparently. And you can see here examples from cyanobacteria, green algae, and plants that are different. They cluster away from all other known rubidoxins. So we have a specific class of these proteins that are essential for photosystems. And Jose, who's in the lab, also has done some biochemical experiments to show that this rubidoxin domain here is exposed to this end of the membrane, the stromal site. So that's also something maybe to keep in mind. Okay, so like I said, you find rubidoxins in cyanobacteria, plants, algae. And interestingly, in cyanobacteria, we have this operon of 6 genes that's conserved through almost all cyanobacteria. And includes this rubidoxin, a known photosystem 2 assembly factor, and then 4 subunits of photosystem 2. So this was pretty good evidence, again, that nature is sort of selecting this rubidoxin for photosystem 2 activity. And Chris already mentioned, too, that there was a little bit of a surprise when we found this because there was some controversy in the literature as to what this rubidoxin was doing. So I was fortunate, I guess, unfortunate enough to go start working in different organisms. So we got a knockout line of Arabidopsis, this flower in the plant. And you can see here that the RBD1 mutant, this is a homozygous mutant, is really, really pitiful. It doesn't grow. This is even with sucrose in the medium. So we're giving it sugar, trying to convince it to grow, and it's not growing. It has no variable fluorescence either, again. And if you look at these photosystem 2 proteins, D1 and D2, there's nothing there. So again, there's a photosystem 2 defect in plants as well. Great. And again, in the interest of overkill, I knocked it out in cyanobacteria. You can see here, so this gene is called RUB-A in cyanobacteria. And we deleted it, and we found that, again, there's a photosystem 2 phenotype. So this is not the most interesting thing in the world. I realize that in all 3 of these organisms, there's a photosystem 2 phenotype, and we delete this rubidoxin. It's less severe in cyanobacteria, but there's still a defect. And we overexpress this protein in the mutant, and we find that it restores photosynthetic abilities. You can see here, variable fluorescence is restored. Okay, so what? So RBD1 is required for photosystem 2 function. That's great, but what it's doing is still not exactly clear. Is it causing protein subunits to be made? Is it interacting with photosystem 2? How is it doing its job? What exactly is this man with the green hat's job? What is he doing? So Chris is a great boss. He bought me a plane ticket to go to Paris, France. It's a lovely place. And apparently I was supposed to do experiments there, so I pieced 2 things real fast and then toured around most of the time. No, that's not true. I was working like crazy. So I went there and what we did was we wanted to look to see, sort of answer some of those questions. So we fed these chlamydomonas mutants radio-labeled acetate, and we saw what was happening at the translational level. And you can see the results here. So we have our wild type. This is a D1 deletion strain, and then this is our Tupac mutant that doesn't have RBD1. And when you feed it radio-labeled acetate, you can see that over 7 minutes at least, the translation or the amount of protein that's getting made in these mutants are all the same. So the RBD1 mutant does not have a translational defect. It's making proteins just as well as the wild type. And so we wanted to say, okay, well maybe there's a stability issue here. So what we did was we took our cells, we added a translational inhibitor, and we watched what happens to steady state levels of the protein over time. So over the course of 4 hours, we can monitor these protein subunit levels via Western blots. And so this is a 10% loading of wild type, that's why it's really low. And you can see here that over the 4-hour experiment, there's basically no degradation or disappearance of any protein subunits. They're all stable. And you can see in this D1 deletion strain, again, there's no D1. But D2, for example, is already very low, and then it's degraded or disappears extremely rapidly. And the same is true for our Tupac mutant. So in the absence of RBD1, these subunits are made, but they disappear extremely quickly. So we also know from some experiments that Jose has done that RBD1 physically interacts with photosystem 2. So he has done some nice co-immunoprecipitations to identify interacting partners, and also some blue native and 2D gels to see which subunits and when photosystem 2 is interacting. And I already mentioned this experiment too, but this is sort of the orientation in the lumen, or in the membranes relative to photosystem 2. So that's sort of the end of the first part. It was pretty quick, I know, but I think it's time to move on to the second part, which again, Chris, well, I think it's a little bit more interesting. Hopefully you do too. So our question was, if we make this interesting double mutant, can we actually sort of trick the cell into making photosystem 2 even though it normally doesn't? So to introduce this idea, I'd like to go back to our little car analogy. We know, again, that we have to have these assembly workers to build a car. And if our little RBD1 guy over here, if his job is to put wheels on the tire, or wheels on the car, if he doesn't show up to work, if he's sick, or if he's just fired or something, we know that you don't just get a car with no wheels. That's ridiculous, right? They don't sell these things to people. That's crazy. And the reason for that is because we have these quality control guys, right? Their job is to sort of hang out and crunch numbers all day and make sure that these cars actually come out and they look like they're supposed to. So he sends this car with no wheels back to the factory floor, and it's like, put some wheels on, you guys get to work. We know also, though, that perhaps after the company party and the holidays, if he's had a little too much to drink, perhaps you can actually get one of these cars with no wheels out, which is obviously not good for the company, not good for anybody. Nobody wins here, except for Homer, I guess. But we know also the same thing is more or less true in the cell. So again, these guys, their job is to do all these fun things in the cell. And we also have a quality control guy in the cell who's making sure that these photosystems are up to snuff and they cut the mustard. It's a weird phrase. We also know that if, for example, we're missing our green shirt, red hat RBD1, quality control says, no mas, this is not cutting it, no photosystem 2 for you today. Fix it and send us a good one. So our natural progression here is to go back to our car factory, and if we can get our quality control guy when we're drunk, we can ask the question of what might come out at the end. And it turns out that we really do know a little bit about quality control, and his name is the FTSH protease. So maybe you guys have heard of this from microbiology or from photosynthesis as well. But the FTSH protease is this really important enzyme in the cell whose job is more or less quality control. If you have a damaged or misfolded or somehow screwy form of an enzyme, FTSH gets rid of it and basically tells the cell, no, do it right this time. And Alizé, who's here, during her PhD actually isolated a mutant of this FTSH, which I consider to be sort of the drunk form of FTSH. And apparently in this mutant, you can actually accumulate these sort of bizarre, misfolded, or misformed protein complexes. Cytochrome B6F was the one that she had studied. So we asked the question, well, can we do this with photosystem 2? So again, I was in Paris, which was fun, and we crossed our Tupac mutant to our FTSH mutant to get this Tupac FTSH double mutant. And we asked the question, if we have this drunk form of the protease, are we getting any more photosystem 2 than we normally had? And yes, it turns out that we have more of these individual subunits. So you can see here, this is Tupac. He doesn't have very much photosystem 2 at all, as we saw earlier. But when you cross it, so FTSH also, I should say, is more or less like wild type under low light. It's indistinguishable. When you cross them, you get this Tupac FTSH double mutant, that accumulates intermediate levels of these protein subunits. So this seems to work slightly. We're getting more of these subunits. But as you guys know from the model and just from common sense, just because you have more subunits doesn't mean you necessarily have more of the protein complex. So Jose and I did a Blue Native 2D gel, and we looked at what's going on inside this double mutant, and we can see that we actually now get photosystem 2 dimers. So you'll have to trust me on here. But these are photosystem 2 dimers that are actually forming in this double mutant. So our leaky protease trick is actually kind of working, which was surprising and interesting. But everything comes with a catch. You know, if you have a car without wheels, it's not going to drive. So the catch here is that this photosystem 2 seems to only be active in the dark, which seems, at least to me, very counterintuitive. Photosystems are supposed to use light to do their job, and if you don't have light present, what's the photosystem doing? And if you turn on the light and it's inactivated, it kind of defeats the purpose. So we found that these photosystems have very low variable fluorescence in the dark, relative to the wild type. And if you expose them to light, it's rapidly degraded. So this is a 6-hour experiment where we're monitoring this variable fluorescence parameter over the course of the experiment. And we have cells that were run in the dark, and we just switch them into light and watch what happens. So you can see here that our highlight conditions, which is 40 microeinstein, which I would say is dimmer than this room right now, which is not really highlight, is causing an inactivation or loss of photosystem 2 after about 4 hours. And even 1 microeinstein, which is darker than below all of your seats, is causing an inactivation of photosystem 2 over the course of 6 hours of about a third, which is really, really strong inactivation and not happening in a wild type cell. So there seems to be something particularly light sensitive about this photosystem 2 that we don't yet really understand. So recently, what I've been trying to do is to purify this photosystem 2 and to figure out what exactly is wrong with it. So for the people in the lab, this is what I've been doing, running around in the dark with these enormous bottles. I'm growing huge amounts of these cultures and then trying to purify a tagged form of this photosystem 2 from the double mutant. And the questions that we're trying to ask are, is this protein's complex missing a subunit? Is it missing CP47 or 43 or whatever? Or is it missing a cofactor? Perhaps this enzyme is required for putting in iron or heme or some metal or some cofactor again. So like I said, I had to cross these mutants using wonderful Metamost genetics tools, which Chris is finally proud of, I think, that I did a cross successfully. And yeah, so we got this 2-POC FTSH, this tag line out, and I purified photosystem 2 from it. And what you can see here is that we actually, maybe you can't really see it, but I can see it on my screen, I promise. You can see that there's photosystem 2 dimers, monomers, and then this small photosystem 2 assembly intermediate that we're pulling out in our wild type cell. And interestingly, in the mutant, we're also pulling out a little bit of photosystem 2 dimer, some photosystem 2 monomer, and some of this intermediate complex too. So this is great. So we have these intact photosystems. What's the difference between the two? Or is there even a difference between the two? And this is called a finishing talk, not a finished talk, so get off my case about it. I don't have an answer yet. I have some preliminary results that I've been doing getting this week and the week before, but I don't really feel comfortable talking about them because I've done it once and it probably is wrong. But basically what I'm trying to do is just figure out what's going on here. What is the difference between these photosystems? And that's what I'm doing. So I'd like to sort of show some models for what we think is happening in the cell. Based on all of the data sort of that we have. I have 2 main hypotheses that we do that we're sort of working on as for what this RBD1 protein is doing. So one of them is this idea that RBD1 is acting as some sort of scaffold or chaperone for the insertion of this non-heme iron in photosystem 2. So again, Jose has shown that RBD1 seems to interact with these PS2 subunits. So perhaps it's interacting here, delivering this sort of red iron atom up here, as photosystem 2 assembles, and then is dissociating or leaving the complex once the iron has been inserted. And this sort of fits with some of the data that we have that suggests that the RBD1 protein isn't part of a mature photosystem 2. Another alternative hypothesis is that RBD1 is acting as a sort of safety valve to recycle electrons back into photosystem 2. And something I should have mentioned but didn't is that photosystem 2 assembly happens in a very stepwise regulated manner. And we know that the machinery required to split water into oxygen is assembled at the very end. But chlorophyll was inserted presumably before that. So what could be happening is you might be absorbing light, driving electron transport, but with nothing to provide electrons into the photosystem, you would probably damage the reaction center and destroy it. So the idea is that this RBD1 protein, which might be an electron transport enzyme, could be recycling enzymes back into the complex before the oxygen evolving machinery has been put together. So hopefully that makes a little sense, and hopefully you guys buy it a little. So I'd like to sort of summarize kind of a little bit of what's going on. So some of you, again, are familiar that Tupac is a rapper, and I want to make sure everybody's sort of familiar with the difference between the rapper and the algo. So I have the species listed here. There's sort of places of birth. The respective bosses. So Tupac, actually, his records were released through Death Row. Ours is released through JVC, a fine journal. These are some famous collaborators I've listed. Tupac also had some problems with the notorious B.I.G. Some of you may have been familiar with that. Our Tupac has some beef with this guy at Penn State, Don Bryant, who some of you again are familiar with. Then there's one other important. This is serious. No, there's one other part that many of you may be familiar with is the presence of LSD. Tupac, the rapper, was fond of this beverage Alizé, which some of you may have tried before. It's really kind of awful. Our Tupac is a bigger fan of this Alizé. I'll know it because she's been helping a lot, and she's pretty awesome too. So then I think I've been doing a lot of other things since I've been here too, and I didn't. Not that I don't have time. It doesn't really all fit together in one coherent story. So I'd like to sort of just mention that briefly in case anybody's interested. But I didn't have time, or not time. I didn't talk a little bit about what Ron, an undergrad who's been working with me, has been doing, which is to try to find other PS2 assembly factors and then knock them out in cyanobacteria. So he's successfully knocked out a gene that we think is a PS2 assembly factor. He's going to be here this summer, so we're hoping to characterize it. I've also been doing a little bit of like very, very basic phylogenetics to try to find genes that are conserved in photosystem 2 containing organisms. So there's this cyanobacterium called UCYNA that doesn't have photosystem 2, and it fixes nitrogen and it lives symbiotically. So I've been using its genome as sort of an outgroup to try to find genes that are strictly conserved in everybody else, but not that one. Something recently that Ron and I have been doing is to try to characterize a deletion strain of this rubidoxin in a thermophilic cyanobacterium with the hopes that we can do some structural work on it and figure out what's going on. I won the Fantasy Baseball League last year. I'm pretty proud of that. That was a big achievement. Yeah, I should probably thank everybody for paying for all of this stuff. HHMI and Gordon and Betty Moore, and Berkeley and the Philomathia Foundation too. But then there's also people. So my family, who flew in, 2/6 of them, I guess, flew in from Indiana, which is really nice. So my parents are here, my sisters are to Hollywood, but they have important jobs and they have to be doing them. My brother's coming in a little bit, and my grandma's here doing her 23andMe kit. That was pretty cool. And I promise I'm almost done with school, so don't worry. Alizé, the QIKO lady. This is not I'm not going to read all this, but you guys can, but yeah, hopefully she's sufficiently embarrassed. Super helpful in all of this stuff. Is a very supportive girlfriend too, and surprisingly normal scientist, which is always pretty cool. I don't mean that offensive to you. Chris, thank you very much for everything. I was looking through my photos on my computer and I found this one. It's funny. This is kind of a funny story. So Chris teaches this biology class, and I'm really happy he mentioned it earlier. We went to Half Moon Bay as like a field trip. We go collect algal samples, and Chris has this like very well planned thing. It was awesome. We were going to go like march around and collect these samples, and apparently we went at the wrong time for like when high tide was coming back in, and so Chris was marching around trying to find the way out, and I'm leading all of these students, like, including Paul, who's here, trying to figure out how to get out, and the tide's rolling in. Chris is on this high cliff, and I have all of the students. Marilyn's there too, and we're all freaking out that we're all going to be washed out to sea. And, so this is at one point, I think, I don't know how I captured this, but this facial expression, I think, from Chris is something that I've seen a lot of, believe it or not. I feel like almost all of the time when I come in and I tell a joke or I try to get him to say something, it's sort of, yeah. But he's been awesome. He's been very supportive, and again, sent me to France to do all these cool experiments, so I can't really complain. My long-lost Spanish cousin, Jose, he's in the back here doing important biochemistry with his lab coat on. Jose's been doing all of the biochemical stuff to try to characterize RVD1, which has been very, very helpful, and he's been a fun colleague to drink beer with, to talk about science with, and overall a pretty nice guy too. And then my long-lost Israeli cousin, Ron. He's the undergrad who's been working in our lab. He's going to be here all this summer. He's not a huge fan of H&S rules, as it turns out, for the long pants and stuff, but he's been very, very diligent and works hard, and he doesn't complain too much. He maybe could be a little more enthusiastic, but that's okay. He does enjoy what he's doing, and it's fun to work with him every day too. I also, in the Mellis lab, there's, I don't know, a lot of you guys know Henning. Henning is a really nice guy. This is funny too. This is a picture that I took. He and I were at a conference in Germany, in Berlin together for Clemena-Monis, and this is Henning obviously taking in the sights, really looking at these beautiful buildings behind him. Working really hard too, but yeah, Henning has been awesome. He works on cyanobacteria too, so I can pick his brain and ask for all sorts of help when I need it, which is fairly often too. And I'd like to thank the NPQ Tang clan, everybody in Chris's lab for showing up to this and for being really helpful too and tolerating me turning off the lights in the middle of the day to keep my samples from degrading and listening to these poor jokes and rap references for the last 5.5 years. Especially Marilyn, who's been helping out a bunch too, very tolerant of my behavior and EHS violating. And everybody else for just being supportive and fun and yeah, good to work with for the last few years. But I'm not leaving, so don't worry. But I'll be around to bother you guys a little bit more. And I didn't mention this too, but our softball team I'd like to thank because we came in second place a couple years ago. It's not quite a championship, but it's pretty good. That's been very fun over the last few years. We've been playing every Sunday night, which has been cool. And then I found this photo of me and my friend Ryan and my parents from, I think, like the second day or third day that we were out here. So this is us. So Thurber was our landlord when we moved here. Now we're both getting theses. Ryan's actually a doctor, I think, officially today, so that's pretty cool. And it's really crazy. It's gone by really quickly, and I'm really happy to have come out here. And I'm sorry that Looney's barbecue closed. The service was pretty terrible, but the food was good. That's life. So yeah, it's been a fun ride, and I'm not done yet. Don't worry. Because I'd like to thank all of the people elsewhere. So Bob, especially, who's on my qualifying exam committee, and it's been fun to talk to about rubidoxins. And I remember talking to him before my qualifying exam about a bar in Indiana called Plum/Shot. It's about the movie Hoosiers, and Bob knew it. I was so happy. This guy's on my qualifying exam. He's so cool. He knows everything about Indiana basketball. We didn't talk about it during the exam, but it was cool. Tasso, who can't be here, he's at a meeting right now. And Jake Healing was my outside, and Peter, who's out of town, but he's been very awesome too. My thesis committee, who again, Tasso can't be here, and Ken's actually out of town too, but Chris is here, so thanks again. And then this photosynthesis journal club, I want to sort of advertise for it. It's been really nice and helpful for the past few years. We have this once a year, and we read papers on photosynthesis, and Chris, Tasso, and Bob put it together, and have been working really hard to keep it going, so that's been really valuable for me too. And then the people who I rotated with, Peter and Jim and Pablo, who's in Spain, so apparently couldn't make it, I understand. These guys were really helpful, and it was really fun to work with them, as well as Shang and Eigen and Shang's lab. And my collaborators again in Paris, these guys are really awesome. It's like a second advisor in some ways. I've been harassing these people with emails constantly, and they respond very quickly, which is surprising and very nice. As well as my collaborators at LEL, Vidal, Junko, and Jan. And there's one more slide, I think. So I also would like to thank Bob Melvin, who's the manager of the Oakland Athletics, because he's done a fantastic job. It was a dark time for Oakland A's baseball before he came here, and he's really done a great job. The Tank Clan, Nicholas, Ryan, and Dustin. Kristen Watford is a former IU basketball player who hit a 3 against the University of Kentucky to win in the last second. It was awesome. So I want to thank him. My rotation, the people who rotated with me, Alex, who's up here, Jason, and Daria, who are here too. That was fun with you guys rotating. Dan Ugla is a current Atlanta Braves player. Jock Jams is what we play at our softball games, and so whoever put that together is a genius. It's really nice. And all these restaurants too. These places have been really good. Speaking of Berkeley, I think it's fair to thank the city of Berkeley because when I moved out here, I felt kind of strange. I don't know, not really that normal, but being in Berkeley has made me feel like the most normal person on the planet. It's really nice. It gives you a lot of self-confidence when you see the hat lady at Berkeley Bowl, especially, who's something else. Yeah, she wears this really crazy bright colored hat, and just seeing her is like, you know what? It's okay. Things are going to be all right. And so if it weren't for her and all the other crazy folks in Berkeley, I think I might be a different person, which may be a good thing. But thanks for Karen, Dana, and Rocio for being really tolerant and helping to keep the nuts and bolts of PNB together and help keep me sort of semi-respectable too. And I don't think any of them are here. I think they're out, but I'll have a second thesis meeting soon. So yeah, and then thanks to all you guys for showing up. I know it's again Friday afternoon, and I really appreciate it. appreciate the support and enthusiasm. So that's it. If you guys have questions, go ahead. Otherwise, we can hang out back and not out back, out in the front, I guess, and have some snacks and some beer and whatever. So thanks again.
All right, I'm sure there's a lot of questions about some of the slides. I know I've got some.
Anything? Yes.
You mentioned some possible mechanisms by which RBD could be impacting photosystem assembly. Do you have some ideas about how the protease actually recognizes?
Yeah, that's a good question. So, so the question is, how does the protease recognize a damaged PS2? How does the quality control process work? And I would probably not be the right person to ask, but I can talk a little bit about what I know. Alizé would know a lot more than I do, but basically, the protease also has another role that I didn't talk about, which is this repair of photosystem 2. And damaged photosystem 2 is more or less cleaned up or fixed by this FTSH protease. And what's thought is that there's cleavage by some proteases, that not the FTSH, in specific parts of the D1 protein, for example, that are releasing fragments into the membrane, which then FTSH actually cleans out. So we see in the FTSH 2POC double mutant accumulation of some of these fragments, even in the dark when there's not photo damage. So we think that these proteases are maybe recognizing, my sort of working hypothesis is that there's a conformational change of the protein where something is exposed that's recognized by the protease, but in a functioning photosystem, it's sort of buried inside the photosystem. Does that make sense? You seem dubious. It's fair. Nothing else? Okay, well, let's go hang out outside. It's nice.